This is part 1 of your 2 cell structure and function videos. Now this video should be a review over what we talked about in class, hypotheses and theories, and also the parts of the microscope. So you might not need to write anything down for this video, but if I say to write it down, then you need to write it down. So a hypothesis is an educated guess, and an experiment is used to test or prove a hypothesis. In order for a hypothesis to be considered testable, it must first be mu. This is a good way to remember it. Measurable, observable, and objective, mu. So what is a theory? A theory is a logical explanation that explains physical and natural events. It is able to be tested by multiple independent researchers. It is well-established, highly reliable explanations that have been tested time and time again. It can change and adapt with new technologies and discoveries as they come about. So make sure you know the difference between a hypothesis and a theory. This is a timeline of what we talked about in class with all those famous scientists. So the first thing that happened was microscopes were invented, then cork cells were observed, then those little animals were observed, then a scientist noticed that all plants are made of cells, then another one noticed that all animals are made of cells, then a paper was published stating that all living things are made of cells, then a scientist hypothesized that all cells have to come from pre-existing cells, and then a scientist disproved the idea of spontaneous generation. Remember we talked about those jars of meat and the flies and the maggots growing? That was the spontaneous generation thing. Parts of a microscope. So, the eyepiece contains the lens to increase magnification, usually by 10 times. So I didn't talk about this too much on Friday, but our microscope has more than one type of lens. There's a lens inside the ocular lens, inside the eyepiece, and there are lenses inside the objectives. So actually, we're magnifying this objective by 10 times. The arm of the microscope supports the objectives and connects them to the stage. You also have to hold it. One of your hands has to be on the arm. The coarse adjustment knob moves the stage up and down, approximately to the correct distance. The fine adjustment knob permits finer focusing by moving the stage in smaller increments. You can't actually see how much the stage is moving. It's such a small movement. The base is the other part that you're supposed to hold when you carry the microscope. The body tube keeps lenses at proper distances from one another. The revolving nosepiece revolves to allow changing the various objectives. And the objectives, we have the 4x, 10x, and 40x magnifies the image. The stage clips hold the slide in place. The stage is where we put the slides. The diaphragm regulates the amount of light going through the stage. The light source passes light through the stage. Now remember, cells are the basic building block of life, and all living things are made of cells. So how do we study cells? Well, we have to use microscopes. Two important characteristics of a microscope that you need to know are magnification and resolution. These are words that you need to be able to define, so you might want to write these down. Magnification is the ability to make an object appear larger. When you are magnifying something, you are making it bigger. So if you use your camera and you are zooming in, you are using magnification. Resolution is the capacity to distinguish between different objects close together. That's a hard definition to understand. When you do take a picture, let's say with your phone, if you zoom in a lot, the picture gets a little bit fuzzy. That's because it's a low resolution. You can't tell the individual little cells or pixels apart when you zoom in too much. But if you have good or high resolution, that means you can tell the difference between different parts of the picture. The microscopes that we use in class are compound microscopes. They use 2 or more lenses to magnify an object. The specimens must be very thin so that we can see them on the microscopes. It has limited magnification, but they are pretty cheap. They are cheap enough that the school can buy microscopes for us to use. Electron microscopes are the other type of microscope. We will not be using electron microscopes. These pass electrons over or through an object to magnify it. They have really good resolution at high magnification. They can zoom way in onto something and you can still see it crystal clear, but they are very expensive, which is why we will not be using them. Microscope use. You're going to need this for Monday and Tuesday. We're going to do a lab. Always start on low magnification, so you use the lowest objective, the shortest one, and coarse adjustment. Then you can move the stage up and down when you're on the lowest magnification. The field of view, which means how much you can see of the slide, is larger on low magnification. And then as you use higher and higher objectives, you are zooming in to a smaller part. Always use the coarse adjustment, the one that moves the stage up and down first, with the lowest power objective. Remember I told you we don't want to put the long objective there and move the stage because we could break the slide or break the lens. We always want the smallest objective and then we can move the stage up and down. Once the specimen is in view, once you can see it on the low objective, then you can switch to a higher objective to see it even better. Magnification of scope. You multiply the eyepiece lens magnification with the objective lens magnification. I sort of gave you an example of this a few minutes ago. The eyepiece magnification is 10 times and the objective magnification is 4 times. What is the total magnification? Well, what's 10 times 4? 40 times. So when we're looking through our 4 by objective, we're actually seeing the object 40 times bigger than it actually is. When we're using our 10x objective, and we multiply that by our 10x lens that's in the eyepiece, what's our total magnification? Well, 10 times 10 is 100 times. So when you're using your 10x objective, it is 100 times bigger than it is in real life. Okay, that is the end of the first video for this unit. You need to also watch part 2 of this video before class Monday and Tuesday. So make sure that you watched part 1 and you've also watched part 2.